Science Unplugged, the podcast where science and conversation collide. Join us for a deeper look into the world of innovation. We are your hosts, Taslima Farouk.
And Charvi Trivedi. Let's explore the insights from the world of science. From the lab to the field, we'll dive into the stories and experiences of researchers, experts, and innovators who are shaping the understanding of our world.
Let's welcome our guest for today, Dr. Dipendu Datta, who is currently a consultant prototype engineer working with Pictura Bio. He did an integrated MSc in systems biology from the University of Hyderabad and then a PhD from the chemical engineering department at IIT Bombay. He moved to the UK as a postdoc at the University of Exeter. Welcome to the show.
Thanks for having me.
So can you tell us what the system biology entail and like how is it different from the other biological sciences like molecular biology and cell biology? So in terms of the research questions that you ask and the approaches that you use to answer these questions?
Sure. So firstly, systems biology is one of those words which has evolved a lot over the years. I first came to hear this term when I joined the integrated MSc course. That was 2009. And that was when it was more of a new word, pretty buzzword then. It was not very clear at that point of time what it was to me, but it promised to say that it was a gateway to interdisciplinary sciences. As in generally, so you gave me the example of cell biology, where you have a very focused term and the scale at which you are studying the biological systems. So you want to study the biological cell itself, its functions, and at that point in time, because the cell is such a small item, you can mostly study molecules which are doing its roles inside it. If you very particularly focus on the molecular interactions, you will get into biophysics, you'll get into molecular biology in terms of its applications, in terms of all of the interactions. That's why physics, the actual usage is molecular biology. Now, if you want to go at a higher scale, when cells come together, that's the tissue, and then organoid. So then it goes on to physiology. For systems biology, you are essentially saying that I'm not going to look at biology in terms of each scale, but I'm going to look at the whole system as a whole. But when you say that, it just makes it a mix of different things, and it can be often unclear what you actually want to do. So the idea comes from other sciences, such as, say, physics, where you look at things in terms of a whole. But even then, you start looking at subjects at their lower levels. So in biology, we started the sciences more of a phenomenological scale. You look at something, you do not know what it is, you study that, you look at parts around it, and you start building a picture. So we essentially have like a dark room where we have small gap pieces of evidence are scattered around, but we do not have an understanding of how each of them are connected. Like we are trying to make a jigsaw puzzle, but we have small pieces, we do not know how to orient them. And given, in order to move forward in biology, to make it a field which is more systematic, where things make more sense, where you understand how these groups interact with each other, an approach was developed, which is the systems biology word. How it comes about is obviously has been evolving in the last 20 years, I would say. Initially, because we had a lot of data around the 2000s when the sequencing became more common, there was a belief in the community that now that sequencing data will be there, people will be able to look at things which are not known about. And from the sequence data, they will be able to understand what's happening. But then they soon realize that you may have the sequence data, sure, but that doesn't give you what is actually happening because of the sequence. What is the functional form of the protein or the other roles, non-protein-based functions of these genes. So we just have been moving from one set of unknown unknowns to a next set of unknown unknowns as we are discovering. But trying to put them across some sort of a framework is largely what systems biology aims to do.
Yeah.
Sorry, go on.
No, you continue, then I'll ask.
No, sure. So that's where systems biology, when I came into systems biology, I was, again, coming from a perspective where originally I wanted to be a physicist, but I had always had an interest in biological questions. As a younger child, I used to collect this newspaper sections which used to have these scientific discoveries into it. And biological discoveries often were interesting to me, but if you look at the Indian system of education where biology comes across to you, you're most likely going to learn about remembering lots of different names, very particular details. It doesn't give you a framework of how you can think about biology compared to other subjects. You don't have any predictive understanding. Like you learn about the names of something, you learn about the anatomy of something, but it doesn't give you an expectation. Oh, I can ask this question. The question-based format is missing in traditional biology. System biology's main approach and philosophy is to kind of change that, make it more like physics, make it more like mathematics, make it more like chemistry, where questions can be looked forward. People can look at the subject and put it in a framework and ask questions. And in order to do that, because biology is so complex, it just requires a multidisciplinary approach. You have to have a certain amount of physics-based modeling understanding, so that if you have these pieces, how do these pieces interact? You have to come up with some expectations that maybe this is happening, but I do not know whether it is happening. Then you can design an experiment potentially to see if this is the way they work. If this increases, does that go down? Or if this increases, that also increases. You start by asking very simple questions like these. If these details match up, then you can make a model out of these things and say that this is the way they are loosely connected. What is the mechanism? I do not know. But once these connections are established, you can ask more directed questions and start building that bridge between these fields and the different subjects. That's largely the philosophy of systems biology.
Right. So you also mentioned about sequencing and modeling, etc. So that is kind of something related to bioinformatics as well. So how does systems biology differ from bioinformatics?
Sure. Again, probably a better way to say it is systems biology is essentially one of those catch-all terms which encompasses multiple different approaches. So within systems biology, there is aspects of bioinformatics, there is aspects of chemical engineering, there is aspects of biophysical modeling, as well as computational and theoretical biology. So if I have, let me take an example. So I don't know if you have heard of this term called flux analysis or metabolic engineering.
Loosely the
Sure.
term. Yes.
Let me just introduce it to you directly. So often bacteria, we know about the biochemical cycles, right? So we know the TCA cycle, glycolysis, all of these things, where essentially a bacteria could take in a certain carbon source, so they can take in sugar, and it will go through these steps where it will break down, and then it will make multiple different metabolites. So in our body also, we are doing similar things. We just don't start from carbon. We just start from more higher metabolites, and then it gets converted into different things which we need. Our body is not able to make all amino acids. That's why we need amino acids from outside. So that's like the essential amino acids, things like that. So inside bacteria, often it is considered, many bacteria can live on their own. They can make all the required metabolites by themselves. So in order to kind of understand what's happening inside bacteria in terms of this metabolic picture, metabolic modeling approaches are used. So you can have the biological details, but then in order to understand what is the kinetics of this, how many, say, sugar molecules per second is the bacteria taking up, and how many alanine is it throwing out at the end. It is such a complicated mess. It's going to be very difficult for you to do this by hand. So people try to make up a model which takes in all of these details together to come up with these kind of answers. But then not all of the details, not all of the information for all of these reactions happening in the cell is known. So what happens instead is you make some sort of a constraint. You make some sort of assumptions what is happening within the cell, and you say that when the bacteria is growing, say at a certain rate per hour, it is eating sugar at certain rate per hour. You make a correlation between the input and the output variable which you can measure, and from that you back calculate potential things which are happening inside. So these are called optimization problems, and that is where like the modeling, the engineering perspective kind of comes into. So at each part, different knowledge groups are required for us to kind of understand different pieces. So in order to set this problem up, to make the metabolic network, put it into a mathematical framework, that is where bioinformatics comes in, which will tell you what are these each individual enzymes which are there, what are the pathways, what are they doing. So set that part up is where bioinformatics comes in. To set the kinetics part of it, to make this mathematical model, that's where computational biology has to come in, where you kind of design it in that way. And you essentially also need a lot of biophysical understanding to define the kinetics. So computational biology leverages biophysical understanding and biochemical understanding to define this. And finally, you have these engineering approaches of optimization to put it all together in this framework, how you can solve this mathematical problem where there is more unknowns than knowns. So each of them are working together, but all of them come together and essentially giving you the understanding of the system.
Thank
So
you
it's
for
basically a
answer.
compilation of everything.
Yeah.
Yes, it is a mess. When you start looking at it initially, you do not know where can you fit in, where can you start. It just because we have segmented it, the biological understanding or fields of biology so much, it feels a lot. But then you can see like it is a question you're interested in, and you always have to use different tools to answer that question. Even as a molecular biologist, today you are going to do some amount of bioinformatics. You're looking at sequence information. You're going to look at, say, a in silico PCR you will do to test whether your primers may be aligned. You're good tests. So you do not think of it as doing like proper bioinformatics because you have segregated yourself as that, oh, this is just a little bit of test I do. It is not bioinformatics in the sense of developing a software. But ultimately, there's a spirit of the tool you are using. So that's where like the idea is like you essentially as a researcher, you're always going to use whatever tool is in front of you, whatever is required to solve the solution, you're going to take it out.
Yeah, that sounds like, yeah, it is messy but fascinating